Hello students, welcome to your biology practical class. Today we are going to discuss about the process of mitosis in case of the onion root tip and in the animal cell. So this is another experiment which is belonging to your spotting experiments. That means you have to just identify and write down which particular stage or which particular type of cell you are observing here. So first we are going to start with the plant cell. So first stage which we are going to identify here is the interphase stage. This is a non-dividing phase of the cell cycle between the two successive cell divisions. Here you will find it out that the chromatin fibers appear in the form of a network within the nucleus. So this is actually the view under the microscope, and this is the simple diagram to understand what is there inside the cell. So as you can see here, the nucleus is very much rounded and it is very much larger in the size. Chromatin networks cannot be identified here. Nuclear envelope, it is clearly visible with the nucleolus. That means all the nucleus parts are present here. Next stage is the prophase stage. So here the chromatin material shortens and condenses into thread-like structures called the chromosome. So these are the chromosome-like structures here. Each chromosome consists of two chromatids jointed at a point called the centromere, and nuclear membrane and nucleolus started disintegration and disappear at the end of the prophase. Prophase is actually been observable into two different phases. This is the early prophase one where the chromosomes are less condensed, while in the late prophase you can find it out, it becomes more larger in the size. Moreover that here the cell membrane is a little bit visible, while the nuclear membrane, sorry, not the cell membrane, the nuclear membrane is present here, but in the late phase you can see that the nuclear membrane is slowly disappearing, and here you cannot figure it out where the nuclear membrane is there. Next stage is the metaphase stage. So in the metaphase stage, a bipolar spindle develops in the cell, chromosome becomes thick, and two chromatids of each chromosome become very much visible. Although in the microscope you can't find it out which one is the chromatid and whether it is been divided into the two portions or not, but the appearance can be seen that all the chromosomes are being there in the equatorial plate, that means in the middle portion of the cell. Secondly, chromosomes become arranged at the equator of the spindle, and each chromosome get attached to the spindle fibers at its centromere. So this is how you can identify the metaphase stage here. Next one is the anaphase stage. Here the two sister chromatids of each chromosome separate from the centromere and move towards the opposite pole. Again, it can be observable into two different phases. One is the early anaphase where just the chromatids have been separated out from each other, while the late anaphase where you will find it out they have taken two polar position inside the same cell, while the spindle apparatus can be visible here also. Last but not the least is the telophase stage. Now in the telophase stage you will find it out that the spindle disappears and the daughter chromosomes uncoil to form chromatin fibers at the two poles. And another thing you can find it out, between the cell there is a thin lining which is the cell plate. It is going to divide the cell into two different portions, and hence the cell division will be completed. Now coming to the animal cells. So first stage in the animal cell is the interphase. The cell contains a prominent nucleus here with intact nuclear membrane and the nucleolus. The nucleolus is not been visible very much in this particular diagram, but the thing is that the nucleus is intact, that is very much observable here. Second thing, chromatin materials appear in the form of the network within the nucleus. Next stage is the prophase stage. Now in the prophase, the chromatin material shortens and condenses into thread-like structures called the chromosome. Each chromosome consists of two chromatids joined at a point called the centromere. The centriole pair replicate to form two pairs of the centriole which migrate towards the opposite poles. Nuclear membrane and nucleolus start disappearing here. So here also you can find it out. This is the early prophase. Both are actually in the early prophase stage. In the late prophase, you will find it out the chromosomes are little visible here inside the cell. And the thing is that at the late prophase, the nuclear membrane will be totally disappeared here. Next stage is the metaphase. So metaphase is very much been identifiable with the help of the arrangement of the chromatids at the equatorial plane, that means at the middle portion of the cell. And you will find it out, the centrioles are now have taken the two polar region position, and hence they have formed the spindle apparatus which have been attached with the middle portions of the chromatids, that means with the centromere region. Next we are coming to the anaphase stage. In the anaphase stage, the sister chromatids of each chromosome separate and appear in the V or I or L in shape, and they are started moving towards the two polar regions. So these two polar regions, these are actually being pointed with the presence of the centrosome, and hence they are moved towards two sides. Last but not the least is the telophase stage. Here you will find it out, the daughter chromosome uncoiled to form the chromatins. Nuclear membrane and nucleoli are reformed to form the daughter nuclei. A constriction start developing. Here you can see in this particular field that the cells are now undergoing an invagination process by which they are going to divide into two diploid daughter cell. So this is how you can identify the telophase stage. So that's it for now. I hope you have understood it well. Thank you.